All right, so here we are back again. Hopefully you guys have out this little sheet here. So it's protein synthesis, it's your labeling sheet. Looking at this particular diagram, you guys hopefully now should be like, hey, that's that particular part. I know what this is. If you are completely lost, I should be able to see you on Google Meet today and probably tomorrow as well, as you guys start formulating more and more questions. Start asking them if you have not. All right, so looking at this particular sheet, like I said, this is an overview of what's going on. So you have parts A through H that are labeled. You guys should be able to go through and look at them and say, okay, that's what I'm looking at. So if this is my cell, it's my animal cell. It's got a cell membrane that goes around it. Inside is my nucleus. I have little openings, which are those nuclear pores. Inside my nucleus, I know that this particular structure, which is letter A here, that particular structure is my DNA, my deoxyribonucleic acid. It is a double helix. Being made out of that DNA, or a segment of that DNA, is letter B. Letter B is a single strand. Letter B is leaving the nucleus. So in order for that to happen, I know that the process of transcription is occurring, and that strand is messenger RNA. It is leaving the nucleus, so it's actually moving in that direction there. It's entering the cytoplasm. When it gets to the cytoplasm, it's going to enter into the ribosome. It's actually going to enter in this way. We can say that's kind of coming down. And as we're looking at it, a couple other things in this cytoplasm, we've got these structures, we've got letter D up here. So let's start with letter C. Letter C, if we go back, that's just the nucleus, and that's what my messenger RNA just left. And then as we're out in that cytoplasm, letter D, these little particles that you guys see up here is what transfer RNA is transporting around the cytoplasm. So what letter D is those amino acids. Remember, those amino acids are my monomers in order to be put together to make my protein. Just real quick, the very beginning of this protein synthesis section, we went through the basic structure of an amino acid. And I said, well, your amino acids kind of always have the same structure. So they're always going to have an amino group, which is that NH2 group, which is this ending here. It's going to have a carboxyl group, the C double bonded to oxygen with an OH group. It's going to have a hydrogen. And my last group, I don't care if it's above or below, these can be reversed, is just what's known as an R group. And that R is a variable. So maybe it is a hydrogen, maybe it's a carbon with hydrogens around it. It is one of 20 endings. And that is the only part of that particular amino acid that's going to change. And like I said, it could be down the bottom, it could be at the top, it doesn't matter. But you always have an amino group, a carboxyl group, a hydrogen, and then whatever else is going to be that R group there. So you have 20 different amino acids. Those amino acids, like I said, are being carried around by your transfer RNA. And they kind of look like little T's there. And on the bottom of that transfer RNA is going to be that anticodon. We'll come back to that and look at it more at the bottom. As transfer RNA brings in those amino acids, it is creating this strand here. This strand that you guys see here is a whole bunch of those amino acids strung together. So we can say, oh, well, it is a polypeptide, which is the same thing as my protein, which is a string of amino acids joined together. So polypeptide and protein mean the same thing. Poly just means many, whole bunch of amino acids joined together. If we're looking at letter G is going to actually point to this large structure that you have here. And that large structure is what's kind of pulling that messenger RNA through and calling out for the appropriate transfer RNA. And that is going to be my ribosome. That ribosome is activated by rRNA. The last part you have is H, and H is referring to your cell membrane. So there's a couple more things. I said circle, label on your particular diagram, and the first thing that you guys should realize is a codon. Know that that codon is in your mRNA. So if this is my messenger RNA strand, I know a codon is every 3 base pairs. I don't care if it's that one, that one, that one. You could have picked the one way down here. But that right there is going to be that codon. My anticodon, however, is on my tRNA. And remember, my anticodon and my codon are complementary to each other. So that structure is that anticodon. Remember, every 3 base pairs in DNA is a triplet. If I'm looking at a peptide bonds, A peptide bond is going to join amino acids. Well, here's a whole bunch of them, but they're not joined together. I can find them joined together on that polypeptide or protein. So I can say, okay, well, in between those two structures right there, that is a peptide bond. You could have maybe gone to this one down here or that one. It does not matter. All of these amino acids are joined by peptide bonds. Next thing says dipeptide. Well, a dipeptide is just two amino acids that are joined together. So as long as you circle two, that's going to represent that dipeptide. So that's kind of what you guys are looking at, is that overall view. So if you're starting to get that big picture, that's great. You guys want to take what you have here. You guys want to fill in what you have for your DNA review. So it says, okay, go through your processes. And we talked about a few. DNA replication. And we also talked about protein synthesis, transcription, and translation. Yesterday, we also talked about going through, or 2 days ago within the video, we talked about, okay, now that we made the protein, how does it get packaged? It's going to travel through the endoplasmic reticulum, get packaged up in the Golgi, it gets secreted, or it gets made into lysosomes. That's kind of what we're looking at here. So it's going to say, okay, what is it? What is it? What is it? Then go through. Where does it happen? Where in the cell does it happen? What part? Protein synthesis, split it up, transcription, translation. What are your steps? And think about the enzymes that we talked about. With this, we didn't really talk about too many enzymes with packaging proteins, so that's okay. The only thing you guys should realize is that lysosomes actually have protein, or have enzymes within them, and those are those digestive enzymes to help them break down old cell parts or food vacuoles or things like that. On the back side, it talks a little bit about what you guys saw on the back page of your diagram notes, and that's basically how viruses can go through this particular process. And we talked about how it has to use that reverse transcriptase and how it can mutate a lot more quickly as they go through that process. So you guys want a little info on that, and make sure you look at that. These are some other little topics that you guys want to be aware of. Anything that you would need, I would make sure that you guys had a codon chart or a wheel there so that you guys would be able to use it. You guys aren't memorizing any of that stuff, but you guys should know how to determine your amino acid utilizing it. Okay, so once again, Google Meet 11:30 today. I'll send that code out. I'll stream it right on Google Classroom a few minutes beforehand if you're interested. If you're like, oh, I can't make it today, I'll do the same thing tomorrow at 9:30. If you want to make both, great. They're not mandatory, but if you have questions, any other questions, just kind of stream them there so that others can see them, and we'll make sure they get answered and go through. Test will be on Friday, so that will be put on Friday morning. You'll have till noon in order to complete that test on Friday on Google Forms, okay? So I'll see you guys in a little bit on Google Meets. Save your questions. Thanks.